This conference will now be recorded.
A very good evening to one and all present here. I hope you all are doing well. I, Bharti Fatnani, on behalf of Biosetup Lifesciences, take this honor to welcome our founder, coordinator, Aragyadeep Das Sir, our resource person, Dr. Soumya Lipsa Rath, faculty members, organizers, and event coordinators. Dr. Kavita Pal, PhD, MBA, Scientific Officer at Advanced Center for Treatment, Research and Education in Cancer, Tata Memorial Center, specialized in nano drug delivery, as well as Biosetup Life Sciences Advisory Member. Dr. Sudeep Das, PhD infection biologist and microbiome expert, Marie Curie European Postdoctoral Fellow, University of Lausanne, Switzerland, and advisory member at Biosetup Life Sciences. Dr. Arpana Parihar, a scientist at CSIR Advanced Materials and Processes Research Institute, Mrs. Somoju Bargavi, MSc PhD, a faculty member at Biosetup Life Sciences, and all my dear student participants. I welcome you all to the webinar on the topic Biomolecular Simulations at the Forefront of Life Science Research, organized by Biosetup Life Sciences. Let me start by introducing our organization, Biosetup Life Sciences. Biosetup Life Sciences is a hub for scientific research and learning approved by ISO 9001 2015 certified and MSME Government of India registered organization having a multidisciplinary team of faculty and research associates that provides support to aspiring students in life sciences. We have successfully organized many workshops, certification courses, career counseling, and competitive exam preparation, such as CSIR, IIT-JAM, GAT-B, IISc, and DBT. We are continuously working for the career upgradation of the students. Without further ado, let me also introduce you to our today's resource person, Dr. Soumya Lipsa Rahat, an inspiring assistant professor from the Department of Biotechnology, NIT Warangal. She has around 12 years of research experience in the field of bioinformatics and has done her PhD from IIT Madras, postdoctoral research at Nagota University, Japan. She specializes in the field of computational biophysics and is inclined toward medically relevant problems. Her lab uses basic to advanced molecular dynamics simulation techniques to explore the structure of protein and protein ligand systems. Her recent COVID-19 HPC consortium project investigates the effect of temperature and humidity on SARS-CoV-2. This was all about the ma'am's introduction. Now, before starting, just a little housekeeping before we get ahead. Please do not share your audio, video, or screen to avoid any kind of disturbance. If you have any questions during the presentation, please type them into the chat box. I'll bring them up after the presentation, and we will also have time for the discussion at the end. Now, I request ma'am to kindly proceed. Over to you, ma'am. Thank you.
Hello, thank you. Am I audible?
Yes, ma'am, you are audible.
Okay, thank you so much for inviting me for this lecture. So, as she said, I'm Dr. Soumya from NIT Warangal. So, I work in the Department of Biotechnology, and today I'll be just discussing a bit about how biomolecular simulations have come at the forefront of life science research these days. Uh, okay. So, when we talk about life sciences, it actually constitutes various fields, and I'm not sure about the audience here, but what I have understood from Argyadeep that it's a mix, right? So, life science also is now you can divide it into various subcategories. We have biomedical sciences, we have plant biotechnology, animal biotechnology, microbiology, food technology, pharmaceutical, agriculture, bioinformatics, etc., etc., etc. I think I have left many aspects, right? So, what is the ultimate aim? What we are trying to do by the life science research, whatever field we are in. So, what is the ultimate aim we are trying to do is to understand how the living cells are actually functioning at the molecular level, right? So what are the mechanisms which is happening, whether how an enzyme is progressing, enzyme activity, or you want to understand the sequence or the structure to provide some new insights or to get a global perspective. So whatever, first thing we do is we try to understand the organism, we try to understand the basics, right? And then what we do is we try to improvise it, we try to improve our living lifestyle, we try to bring like we have GM crops, we have transgenic animals, all these things, right? So this is the ultimate aim what we are trying to do in life sciences, right? Now, what happened is initially that when this field was progressing, when people were researching on like different areas of biology, first thing came was a protein, not the DNA. So first people tried to first people got information about the protein, about the structure of a protein. So protein was crystallized, and many years later, people started sequencing the DNA, right? And so we have different several techniques for DNA sequencing, and now with the next generation sequencing we have an explosive growth of biological information. There is a lot of data. Similarly, now we have crystallization, we have cryo-EM, and we also have NMR. You get information about the protein structure, sequence. We have information about the DNA structure, DNA sequence, and we also have many more information available now in the form of cDNA, mRNA, and we have small molecule which are approved by FDA, or we have a like chemical ligand database, we have PubChem and all that. So many information is available to us. So what we need to do is we need to store them, right? We need to store them so that we can manage them. And once we are able to store, we are able to manage, we should also be able to retrieve some information from that data. We are doing experiments, we're getting some results, we'll store it, but from that results also we should try to extract as much information as possible. And this data also needs to be updated regularly, and it should be freely available for researchers like us so that we can do our research properly. Okay. So that was about how the bioinformatics field came up. But gradually, people saw that actually there is a difference between bioinformatics and computational biology. So bioinformatics is what? You have large set of biodata, biological statistics, you have results of scientific studies. So that data storing, data managing, data processing, majorly that is what is bioinformatics. But when we talk about computational biology, it is concerned with the solutions to issues which are raised by studies in bioinformatics. So suppose I am thinking that, okay, what is happening if there is a mutation in a protein? So I am seeing there is a structural change, I am seeing there is a sequence change, but so what? What is the net effect? Okay, if I have to address questions like that, so what I'll do is I'll do research on computers, but that is not bioinformatics, that is a separate area, it's called computational biology. And computational biology, it is very useful in scientific research, including how you examine how the proteins are interacting with each other. So we have heard about enzyme substrate complex, but how do they interact? We know, okay, there are weak interactions which reduce the activation energy and all that. So what exactly is happening? Is there a role of any ion? Is there a role of water molecule? So all that we can study by using computational biology tools. Okay, we can also see folding of a protein. This was not possible experimentally. So how do you see folding of a protein in lab? It's very difficult, but it is now possible to see that on a computer. You can see how the dynamics, the proteins, and say for example, we ourselves, we are not very static systems, right? We are very dynamic. There is lot of movement happening inside our body, in our brains. So it's a very dynamic system. Similarly, proteins are also in motion all the time. So how these proteins dynamics and how do they interact with other molecules, with themselves, I mean with similar protein interaction, protein lipid or any carbohydrate interaction, and what is the net result? All this you can study by using computational biology. And there is, before I start, I want to make clear the term simulation and stimulation. So often people get confused with these two terms, simulation and stimulation. So stimulation is STI, stimulation meaning you're making an organism to act. Say for example, here a person is sitting on a wheelchair and you're giving an electrical impulse, so maybe to like trigger the muscle or something. So that is called stimulation, or stimulating, right? But simulation, SIM, it means to imitate something. So what we are doing here in simulation is we are trying to mimic what is existing in a system. Like here on the right side you can see there is a weather forecast. Okay, so weather forecast is not exact actually happening, but we are predicting ki, okay, today it might rain, or today there will be even the temperature is even going to rise, right? So this is all prediction. So there is a difference between simulation and stimulation. So what we are going to see today is majorly about simulation. So how do we set up a simulation? So simulation, as I said, it's a mimicking. So it's a real world system we have, and or a physical model. So what is happening is we're trying to imitate that, and we're trying to get more information from that. So maybe that is not happening at that instant, but we can get some information about what is the weather, 10 days weather report we can get. Okay. So these imitations, they are made on a computer, and usually what happens is first you build a system. So the you build a model, and this model is nothing but some mathematical representation, and this representation of a real world system, and you put it in the form of a computer program. And in this program you also add few theories, few approximations, because of course it's a model system, it's not a real system, so everything won't match. Okay, and on this model you do simulation, or you do some experiments, and you get some results. These results, what we first do is you compare it with those in the experiments. So you need to verify and validate that whatever theory approximation you have taken, whatever model you have built, it is matching with what is there in experiments. Okay, so whatever you're predicting in a theory, it is matching with the experiment. If it is matching, then okay, we are successful. We can go ahead with further experiments. We can compare even more things, right? We can see more parameters, and we can examine what will happen to maybe some other factor you can see. But if it is failing, what we do is we go back and we again try to uh, fine tune the theories, or check the approximations again. So this simulation is majorly used in weather forecasting, in car designing, car crash simulation. So what happens when a car is crashing? So what will be the next step? So that you can see building architecture, bridge simulation, aeronautics, building stability, life sciences. In all these areas you have simulations. What is the advantage of simulation? it is safe, so you're not harming a person, you're not harming any environment, and it's very safe. You are predicting predictions. It will help us to predict the future course of a particular event. It is very economic because it is saving time and money for us, which is spent in any experiment. And visualization, we can visualize those events also, so you see on a computer how the change is happening. So when we see something, it's very grasp the information more. And replication, so if you are not sure, if say, if the a protein is behaving like this at this particular temperature, what happens if I change the environment, if I increase the temperature? So the same setup, you just change one parameter and see how it is behaving. So you can also do it more than once and you'll get the similar results. So those things are possible in simulation. And what are the disadvantages? There is theory and there is approximation. So a lot of assumptions are taken into account when we are setting up the system. So that is one disadvantage. And another disadvantage is you require very high power for few of the systems. So few systems, very huge systems, or if there are a lot of theories, lot of mathematical equations, it will require time, which is not possible on a laptop or a normal desktop. Sometimes it might require supercomputers. Okay. So what are the different questions in life sciences which we ask? So generally we want to see the rate of a reaction, we want to see enzyme inhibition, we want to see what is the effect of mutation, effect of small molecule inhibitors, how do they act, protein folding or free energy of binding, solvation, interaction, all this. You can check the diffusion coefficient and many more questions. So each lab has a different set of questions. They're doing different experiments worldwide. Okay. So all these questions are at the level of either proteins or maybe so they, they start at the protein level. Yes. Any doubts? Okay. Right. So they start at the protein level, or you can go to small organelles or even complete cell. Right. So how far have we reached in simulation? So we are now able to do simulation of atoms, small molecules, proteins, or even a membrane bilayer or carbohydrate moieties, DNA. We can do simulation of that. We can also simulate a virus capsid or small organelles. It is possible, but we haven't reached so far as bacterium is concerned. Okay. So if we go to this second line here, the purple color one, at the level of atoms or small molecules, when we do simulation, what we are doing is we are taking the electronic configuration and we are trying to get the details about maybe the charge and the map and the like how they are distributed and what is the concentration, the electron cloud and all that. So you need quantum mechanical simulation for that. It is at the quantum level. When we go a little bit higher, so maybe protein ligand interaction, protein folding, protein and like lipid or protein whatever, so that is called all atom molecular dynamics simulation. Here what we do is there is an approximation and theory involved in this step, where we are losing few of the details which are there in the quantum mechanical simulation. Okay. So whatever this was very detailed, slowly we are losing the details and we are leaving out few points. We are not taking into account electrons in this level. Okay. So it is just atoms, and sometimes you take charge, or it just you don't take into uh, very detailed explanation of the atoms. Okay. So this is all atom molecular dynamics, but you are able to get the overall result. Then comes virus capsid and small organelles. So here even more approximation. So these are called coarse grained molecular dynamic simulation. What happens is you take few atoms, group them together, you make them coarse grained. Okay. So you can get the information of the overall structure, but you lose a lot of details in that. You don't get atom level information, you don't get electronic level information. So this is coarse grained MD simulation. Okay. And when we are talking about the dynamics of a system, in atoms, so sorry, the size of the system. So atoms are around 1 angstrom, small molecules 1 nanometer, proteins are around 10 nanometers, whereas virus capsids 100, and bacterium, if you see, it's 1 micrometer. So gradually the size of the system is increasing. As the size of the system increases, the complexity of the system increases. So it depends on what you want to study. You choose your kind of simulation you want to do. Okay. So again, so biomolecular simulation, majorly three parts: quantum, all atom molecular dynamics simulation, or coarse grained MD simulation. So these three I have told you. So in the quantum level, you have atoms, electrons, clouds are included, explicit solvents, quantum mechanics, all this comes here. You have all atom MD simulation where all or the most or most atoms are present. Explicit solvent, meaning water is present all around, and you do MD simulation in this. This is quantum mechanics, this is molecular dynamics. Coarse grained molecular dynamics, what you do here is you group different atoms together. So they are beads composed of few atoms. You can have explicit or implicit solvent. Explicit meaning water molecules are present, we can see them. Implicit meaning, imagine there is a water box around, but it's but you can't see them. Okay. Then you can even coarse, make it more coarse, so supra coarse grained. So here what happens, the interaction sites comprising of many atoms, protein parts or proteins, they are made into huge balls. Okay, the supra coarse grained. And this is an implicit solvent, and it's called stochastic dynamics. Or you can have a continuum system where everything is like a continuous mass, implicit solvent, so you lose out a lot of details in the last step. Okay. So majorly what people do nowadays, so if people are from chemistry, or if they want to study small molecules, generally they resort to quantum mechanics. And biological sciences people generally do all atom or coarse grained simulation. And I think very few people do supra coarse grained or continuum simulations. So this part constitutes a major chunk of research these days. So how did it start? So you'll think, when did it start? How did it all start? So first, what happened? People started to study interaction of two simple gas molecules. Okay, so gas molecules, two molecules of gas, you just try to bring them together. So what happens if you bring them together? They will have some sort of attraction, but after a point there will be some repulsion. So, and if there are two atoms, they can also have electrostatic interaction, they can have hydrogen bonds, they can have covalent interaction. So all these different criteria people started to study. They made a model, they uh, tried to verify it with experiments, and they found that okay, it is matching. Okay. Then in 1977, McCammon et al. He performed the first MD simulation of a protein. So this was the first protein simulation, and this was the smallest protein. And so if you see, this is structure A and structure B. So this change is happening. There are very few changes around maybe this place here. So what is happening is is after 3.2 picoseconds, which is very very less time. Okay. And now so that time the computer was not very advanced. Now we have many supercomputers available, and we can run microsecond simulations also in a day. Okay, so we have advanced a lot. And when we are talking about the time scale, so quantum QM simulation of electrons. So if you do it, it's in the range of femtoseconds. Molecular dynamics, you do nanoseconds to microsecond simulation. Coarse grained simulation, you can do it further till seconds, but you lose the details, of course. So molecular dynamics, basically, it is happening in the range of nanoseconds to microseconds. Okay, and this much time is enough if you want to see a bond formation, a change of alpha helix to beta sheet, or you want to see some significant biological change, you can observe it using molecular dynamic simulation. Right. So, so you will say that, okay, why do we do simulation? Why does the structure change after some time? Because, as I said, our system is very dynamic. So everything is in motion in our body, inside the cell, in the proteins. Everything is in motion. So when we do a simulation, what we are doing is we are trying to see that protein, and we are seeing how is it changing over a period of time if we give that particular environment. So here, are we going to push or pull the protein? No, we are not doing that. The protein is moving on its own, okay, because they are very dynamic structures. So why are they dynamic? In lab, when we do an experiment, so you take a flask and you put a solution. In that solution you have a protein. It is in at a particular pH, at a particular temperature. You have few ions there, right? So similarly, we just mimic that on a computer. We have a molecular simulation, we have a water box. So these all are water, this is a protein, and we can have few ions here. We can mimic that particular temperature, that pressure, and pH, everything we can do. So, and this box is like completely surrounding the protein, and it is also like periodically distributed all around. So it's a periodic boundary condition, and so all around you think that it is inside a water box, the protein. So whatever change is happening, so suppose if you put an inhibitor here or a drug molecule, how the protein is interacting with that drug molecule, that you can see. Okay. Here what is happening is, okay, fine, this is happening, this is changing, but how does the computer calculate how it is changing? That is another question which comes into our mind. So what we are doing is, this is at the macroscopic level. Macroscopic meaning large scale. What we are seeing on a computer is a microscopic level, which is only one protein. When we take a solution having protein, it is not a single protein. You can have different proteins, and you do the experiment 2 3 times. You take a standard deviation to see the behavior of maybe 2 3 replicas. But what we are doing here is a single protein. So you're seeing the observation of at the microscopic level, and we are trying to relate the microscopic level observation to the macroscopic level observation by using some statistical mechanical techniques, statistical mechanics. Okay. So just let me show you the structure of HIV virus capsid. Very beautifully done. So this is the complete virus structure. It's a coarse grained model, and here you can see blue color, green color, red color structures here. And this is the water box with lot of ions around. So small dots, if you can see, yellow and cyan color dots, these are surrounding the virus. Okay. And now if we go inside, we can see the structure. Like, what is this blue color? It is actually the protein. It's a capsid protein. How is it folded, and how does it make this flower like structure? That we can see. So we can also see the pore, which might be allowing the passage of water and ions inside the capsid, right? So how do these molecules interact? So once the system is set, how do they interact and what effect it has on the system? That is the question which comes into our mind. So the molecules can have either bonded or non-bonded interaction. Bonded in the sense, if two atoms are very close by, they are bonded to each other. then they will be having some interactions. And since you can see here, they are moving, they're dynamic structures, so they will be having some flexibility, and there will be some bond energy associated with it. Okay. Similarly, you have angles. So in angles also, one, two, three. You have three atoms which are connected. In dihedrals, you have four. So these are bonded interactions. So each bonded interaction, you will have a certain, like you have a, a range in which the particular bond can stretch, or angle can bend, or torsion can move. So that particular range and energy, there will be like a spring. It's like a spring. You can stretch a spring for this long. You cannot stretch it further because it will break. So similarly, we have some limitation, that is energy associated with that. So that is there. And we also have non-bonded interactions where we have maybe electrostatic interactions, which it is making with the ions or maybe water molecules. So those interactions are happening inside the cell. So you have bonded interaction, bond angles, dihedral, non-bonded interaction, van der Waals, and electrostatic interactions. So these sort of interactions, what they do is they will give you an energy. So this energy is the potential energy of the system. So how do these molecules interact and what effect it has on the system? To know that, the potential energy is calculated by these interactions. The potential energy is the energy of an object or a system due to the position of the body or arrangement of particles in the system. So in the first case, we take a protein, put it in a water box, take ions, whatever you make the setup. Then after that, there is some function which is given. So this function is like MD functions, whatever we give them as initial velocity, random velocity is given, and as a result of which the system starts to become dynamic. In the end, what you get, a resultant energy, which is the result of the position of the body and the arrangements of the particles in the system. So, and the energy, as I said, is a function of atomic positions R of all the atoms in the system. These atomic positions, they are expressed in terms of the Cartesian coordinates. Cartesian coordinates meaning position of any place, you can say from the XYZ coordinates. Similarly, for each atom, you have XYZ coordinates. You know the bonded energy, you have the non-bonded energy. You calculate the potential energy of the system. And what is the nature of a protein? The nature of a protein is to be in the minimum energy configuration for its optimal activity. So that is how you calculate the potential energy of the system. Yeah, so yeah, this is the just a layman description of MD. So MD simulation is a molecular dynamic simulation. It's a computational tool to explore evolution of a protein over a period of time. You do the simulation in a near cellular environment, and you take a solution phase, maybe around 300 to 310 Kelvin temperature, and you do the simulation generally in at the atomistic level. What we do here is we solve the Newton's equation of motion, which is F is equal to ma, and we relate it to the negative gradient of potential energy. So this is F is equal to ma. A is nothing but the position of atoms here, and so we can just put everything with respect to the position of atoms, and we can relate this force as the negative potential of negative gradient of potential energy. Okay, and we can relate both of them, the potential energy and the position of atoms. So I'll show few case studies. Yeah, so this is one. It's not responding. Yeah. Hmm. It's not responding. Okay. So not sure why it's not working. Let me see. Yes, yeah. So this is you take a drop of water on a hydrophobic surface. Okay, so if you see, this is the stable part is a hydrophobic surface, and these are water molecules. So it starts with a square water box, and gradually, if you see, due to the surface tension, it becomes spherical. It's like, so that change is seen in the simulation. How over a period of time it changes, right? Yeah. So if you see it here, it starts like a box of water, and in the end, it becomes like a complete sphere. Okay. Similarly, I'll show you another example. So here, okay, this has a problem. So what exactly was happening here is there was a pulling of DNA in this. So this is a membrane, and this is a membrane protein, and this DNA is pulled inside. Okay, so that pulling inside of this particular orange color DNA by the protein over a period of time, that could be seen. And this is another example where here also you see a membrane protein, and this is the membrane. So protein is deleted here, and on the right, on the top, you can see the time frame. So how gradually over a period of time the membrane is sort of bending. Okay, and now if you see the structure, there is some structural change also happening. So how is it bending? It's not bending symmetrically, it's bending a little bit angled. Okay, these are a few examples. So some remarkable findings from MD simulation. Uh, so this is the structure of H1N1 virus. So this complete model was built. We have various protein structures now available. So on the surface we have receptors of the virus which interact with the proteins on the of the virus which interact with human receptors. Like you have a spike protein for SARS-CoV-2. Similarly, here also you have various receptor glycoproteins and all that. So those this is a coarse grained model. Okay, and it's a very beautiful model, and the so you can see how it is changing over a period of time, how it is interacting. And on the right side, this is a folding of a protein. If you start from completely unfolded structure, you can see, you can make the whole energy landscape, free energy landscape. So this is one metastable state, this is another stable state, another stable state, another stable state, and this is the most stable conformation where the completely folded structure of the protein is seen. Okay. Similarly, like we are doing docking, right? Molecular docking now everyone does, but is that stable for long period of time? We don't know. So for that, what we need to see is, we want to see, like we'll run the simulation for few nanoseconds. If the drug is still staying there in the pocket, we'll say okay, fine, it is happy there, let it sit there, it's okay. But what happens is, in many cases the drug might not stay there, it will just come out after some time, due to the maybe solvent effect, or maybe some side chains are obstructing it to stay for longer time once they're dynamic. So all that is seen here. Okay, so so these are the different interactions also which we have seen earlier in you might have known from docking, but then what happens when MD simulation happens? Is there any extra interaction we are seeing, or is it lost? That sort of things we can see. Similarly, here this is the structure of a DNA. The DNA has a very beautiful spine of hydration, and many times what happens, people design inhibitors or molecules to store for a longer period of time. So these molecules, they come and sit in the spine of hydration, removing the water molecules but absolutely preserving the structure of DNA. So that at the molecular level, how you can see, we can see that in a simulation. So this is a bDNA, and you see how the structure is maintained here. So if the structure is not maintained, what happens? We don't want that, right? So we can check that in computers and then go for experiments. This is another example how you the proteins are helping in the bending of a membrane. So these are bar membranes, okay, sorry, bar proteins, and they help in the folding of a membrane. So I'll just show a few examples. Yeah. So this is how it happens. So you see the red color, yellow and green, blue, all these are different proteins. So over a period of time, which is here in the nanoseconds, here it is shown. So it is completely, after some time you see it becomes completely tube-like. This is happening inside our body, and now we can see it on a computer. That is the beauty of computer simulation. Yeah, so it is completely folded now, right? Like a tube. Perhaps the most crucial application of MD simulation during this COVID-19 has been its ability to reveal the structural dynamics and the arrangements of viral proteins and associated protein-protein interactions. So it's very difficult when a pandemic like this comes, right? All of a sudden it comes and all the labs are closed. We cannot do any experiments, and the virus is very dangerous, right? So we need particular setup, some safety levels are required to do experiments. So what do you do at that time? We can use our knowledge of computer simulations, modeling and docking to get some more information before people go for experiments. So I think there were a lot of computational studies during this COVID, when there was a shutdown, and so major studies were done on the spike glycoprotein. So as you can see here, the structure of the spike glycoprotein, the red color ones. So it is one of the most prominent structures of SARS-CoV-2. It's present on the surface of the virus envelope and helps in the attachment to the human receptor. Okay. So how it acts is, you have the spike protein and then there is a receptor. So this spike protein and receptor, they have an interaction, and then there is this furin which comes and it cuts it off. Okay. So once it cuts it off, then the remaining part, it can make a tunnel sort of thing from the viral membrane to the host membrane to transfer materials um, inside, right? So they can communicate very easily. And over a period of time, there were many variants like alpha, beta, delta, kappa, omicron. Many variants were had also come. So they were also studied in the on the computer initially. And so what we got to know from this computer simulation, first thing we got to know is that there were three states actually of the SARS-CoV. There were open, or they can be semi-open, or they can be closed. So this is like an open state where you see the flaps here open. This is closed. Sometimes it will be half open. Okay. Or like here you can see the effect of temperature. What happens when there is a increase in temperature? At low temperature, it is the spike protein on the top. If you see this part here from the, like, if you look it from the top, okay, you see it is sort of open, but when you increase the temperature, it closely comes together, shutting its it's like very tightly and very closed compact structure, where it is actually hiding the interaction interacting regions. Okay. So that is another study, and this is a study where we have seen the effect of temperature on the membrane protein of SARS-CoV-2. So these are the membrane proteins are also present. So here, if you see, apart from the spike protein, very small proteins are also present. These are the membrane proteins, and they also help in the formation of a membrane structure. Okay, the virus capsid and all, sorry, envelope. Okay, so if you see the effect of temperature, then what happens is as you increase the temperature, they are actually aggregating and they are sort of becoming very flexible, the membrane, when you increase, and they're also inducing some sort of curvature on the structure. This is another paper where they have seen that it is actually this glycoprotein, the glycans, the carbohydrate moieties on the surface of the virus. It is heavily carbohydrate, I mean glycosylated, sorry, glycosylated. And what happens is this blue color structure here, they have seen, so Amaro et al. They performed microsecond MD simulation on the fully glycosylated S protein, and they identified that this particular domain, so this domain which is actually interacting with the ACE2 receptor, and they play a significant role in the S protein binding and dynamics, they have actually very less glycans. So that is a very important finding if you want to design some antibody or vaccine for a virus. So these molecular details are now they are very important, and it was very helpful for researchers all around. Also, they could compare what exactly happened between SARS-CoV and SARS-CoV-2. What they found was there was an increasing interaction energy between the human ACE2 receptor and SARS, the receptor binding domain, the spike. So as there was a little bit mutation in SARS-CoV-2, and that mutation actually increased the binding energy, so it was even more strongly binding. Then there were also research on non-structural proteins like protease and other like the effect of mutation, protein interaction. All these have been done. Yeah, so this is how the mutants. Then various variants have come. What exactly happens if there is a mutation? What is the energy difference? That was also seen, and of course these new variants slowly crept up, and it was found that actually they're changing the total binding energy of the protein with the human receptor, and there is also distribution of charge is also changing. So that makes a lot of difference in the protein protein interaction. And this was one which is with a few microseconds, and how the inhibitors are interacting with the spike. These are FDA approved drugs, and how do they interact over a few microseconds. So this is for 2 microseconds. This is another, yeah, the same one. So at the microscopic level, at the molecular level, what is exactly happening? So this was one. This is another inhibitor which is binding at a different site. What happens if it binds to another region which is not at the center of the three monomers of the spike? You can see here how it is bonded. And is the bond stable? Yes, it is stable for 2 microseconds. So it is going to be stable for long time. Similarly, a bigger inhibitor structure. So this is binding to only one of the spike protein monomer. Here also there is a stable bond which was found, and a lot of hydrophobic residues. So that sort of details could be seen very easily. Right. So for doing MD simulation, what you need is a model, whether your protein or protein ligand, DNA, RNA, lipid, or small molecules, whatever. You need some softwares. Commonly used softwares are Amber, Gromax, NAMD, LAMS, CHARM, and you need to know the information about the system, like how they are connected, the bonds, angle, dihedral, and all that. So you need to choose a particular force field for that, which depends on what you want to study. So after you set up the system, you do a minimization, you minimize the system, you equilibrate, and you run a production. So what is a minimization? You optimize the geometry. So you try to bring it to a low energy, like optimize the geometry in a very minimum energy conformation. Then you do a equilibration. So depending on what environment you want to keep it in, whether it's NVT, NPT, or NVE, whatever, so you give that environment, you try to tell the system to relax there. Okay, and then you let it run, you let it evolve over a period of time, maybe nanoseconds or microseconds. And of course large system, you need large computational resources, so it depends on what you want to study. So with that, I think I'll end my talk. So I'll end it with the saying of Richard Feynman that everything that living things do can be understood in terms of jiggling and wiggling of atoms. So that's all. Thank you. Any questions?
Thank you so much, ma'am, for your insights and knowledge onto this most important topic. We will go ahead and take some time for questions now. So, any student having any questions can drop your questions into the chat box.
Ma'am,
Okay,
please
so
explain about the folding of villine protein in a simulated folding pathway. So you want to know about this protein folding? Okay, so something like this maybe. So if you see here, villine protein. So if you have a, so it will when you fold a protein, it goes through a energy this funnel, right? So it will be there. It's a structure is more stable. The so energy will be minimum in this funnel. So suppose this is a villine protein. I'm not sure if this is the structure. So what happens is it will make some interactions. Okay, but that interaction is only transient for some time. After that it again breaks, so it will form another structure. It goes to another metastable state, stays for maybe a little bit longer time, again it breaks. But when it comes to structure like this where a lot of interactions are there, it is no longer breaking. It means that this is a very stable structure. So villine also, I the general protein folding happens in the similar way. Did I answer you? Did I answer your question, Mr. Sayankar?
Yes, ma'am. That's right, ma'am.
Okay. Ma'am, it is used in drug discovery. No, it's not used in drug discovery. Drug discovery, you will use maybe virtual screening, molecular docking, and structure based drug discovery. All that is different. But molecular dynamics, if you want to validate, okay, the drug is still binding for a longer time, it is staying there for a long time. Then you use it, then you use simulation. What happens when you keep the system like that for longer time? Ma'am, do they find the free energy of the system the way of how Williams structure diagram is? Yes, we do find there are different. You can find actually free energy landscape. You can do how the stable 3D model of a protein is made from revolving protein molecule. So actually this is not a, it is a revolving molecule. What I have shown you only is the 2D structure. So we have visualization tools like PyMol and VMD and all that, Rasmol and all that. So you can rotate the molecule. So that position is XYZ coordinates. They give you the position of each atom, and based on that the 3D model of the protein is made, and you can change it. You rotate it along those axes: X axis, Y axis, Z axis.
Do DJ. Do DJ. Good evening, ma'am.
Yeah.
I'm Sujit Power from Solapur.
Okay. Yes.
Which department is used? That is biochemistry from cell biology.
No, actually, this will. This is not there in all the. This is under bioinformatics. So it is a part of
Only
biotechnology. and a sub part
No,
of bioinformatics. But yeah,
only bioinformatics, that is computational methods.
yes, computational biology.
Okay, no used in the biochemistry and laboratory studies.
No, lab studies are different. What we are doing in experiments, wet lab studies, that is different, biochemistry and all. So this is all computational study, but we are trying to answer few questions which, so like if see protein folding, you cannot see it on a lab. So you can use computers for that.
Then how it is show that the protein folding will be changed or not the changed.
We'll use some computer simulation. So we are mimicking the system. We'll see its evolution over a period of time. After a period of time, what change is happening that we are seeing on a computer.
Okay, thank you ma'am, bye.
Okay.
Okay. Thank you so much, ma'am, for answering all the questions. It looks like we have covered all the questions now. So if there are no questions anymore, I would request Aragyadeep Das sir to propose the vote of thanks. Over to you, sir. Thank you.
Am I audible?
Yes, sir.
Yes, sir.
Okay.
Yes sir.
Very good evening to one and all. My heartfelt thanks to our eminent guest speaker, Dr. Soumya Lipsa Rath, and I thank you for accepting my invitation for the interactive talk and come webinar. So it was an interactive and research-oriented talk from a great woman scientist. And you know, ma'am, the topic of your collection is so useful for the students and researchers community nowadays. And day by day, bioinformatics, computational biology scopes are increases, and because without bioinformatics stuffs and research is not possible. So people are actually looking forward for this type of topics, and the main important thing is that you share your knowledge in this topic was so impressive, ma'am. And uh, the topic is so vast, and in 1 hour talk you will give insight knowledge in very concise way. It was a remarkable and miraculous experience. Literally I was enjoying it a lot, and I learned a lot of things to boost up my knowledge. And ma'am, I hope all are liking it too. So thank you for sparing your precious time and share your knowledge with us, ma'am. And a big thank you for answering all the questions and interact with the students and researchers community, uh, and be a patience. And it was a really a great experience, ma'am. And it's
Thank
given
you
me
so
a
much.
immense pleasure. Yeah, thank you, ma'am. It's given me a immense pleasure to deliver vote of thanks to our today's eminent guest speaker, Dr. Soma Lipsa, and also all the participants. Once again, I thank you, ma'am, for such an interesting research-oriented talk, and your dedication and your work in field, not only the science and also the betterment scientific society, helping nature is really a commendable and attractive, ma'am. And
Thank you so
even
much.
you are you are also working for awaiting people during this pandemic, and you are awaiting with the help of news channel in media. I saw your couple of interviews, and ma'am, it was a rememberable and experience. Hats off to you, ma'am, as a woman scientist, and a long journey of your research. It's so impressive,
Thank you so
and
much.
I'm looking forward for your guidance in future too. Thank you once again, ma'am. And
Thank you. Thank you so
now
much for
my
so many words for me. So yeah, thanks a lot. Thank you.
thanks to our organizer, event coordinator Dr. Kavita Pal, Dr. Arpana Parihar, Dr. Das, and Miss Somuju Bhargavi, and our organizer Miss Sneha Mahajan, Ananya, and Shreyans, student volunteers, Bharti Patnani for wonderful anchoring and welcoming us. And all my dear students and researchers, thank you for joining and invest your precious time for listening to the talk and support us. Last but not the least, on the behalf of Biosetup Life Sciences, I thank each and everyone for contribution to make this webinar a successful. And I'm looking forward for arranging many such events. Thank you all. Stay connected with us. Good night. Thank you, ma'am.
Thank you, sir. Thank you, everyone. We appreciate you being
Thank you
here.
again.
For the students, filling out the feedback form is mandatory for your attendance. The feedback link has been shared in the chat box. Thanks again for joining us today. Hope to see you next time again. Thank you.